Today we're going to be covering Chapter 1, Biology, the Study of Life. Now, I am going to warn you, it's a pretty long PowerPoint, but what I want you guys to do is, obviously, you're going to be taking notes off of anything that I underline, and if you can't get it all done at once, that's fine. You can always just pause and then, come back later. Just go ahead and mark down where you left off on which slide so that you can come back and finish the remainder of it. I don't expect you guys to get it all done in one shot, but let's go ahead and get started. So we are going to cover 6 parts, and we're going to start off with part 1. So what does it mean to say that something is alive? So if something is living, it's going to share 5 fundamental characteristics. The very first one is cells. So everything is going to be composed of membrane-bound cells. So what that means is it just basically is going to have a membrane that surrounds them and keeps them completely separate from everything else. Okay, that does include prokaryotes and eukaryotes. So if by any chance you don't remember what prokaryotes and eukaryotes are, I would suggest you write it down. But prokaryotes are not going to contain a nucleus, whereas eukaryotes do possess a nucleus. And this is what a prokaryote is. So mainly the only thing that's going to be a prokaryote are bacterial cells. And then eukaryotes can be anything else. So it's going to be protists, plants, animals, and fungus. Number 2 is replication. All organisms are capable of reproducing. Number 3 is information. All organisms are going to process hereditary information that's going to be found in your genes, as well as information from the environment. Number 4 is energy. All organisms are going to acquire and use energy to stay alive. Number 5 is evolution. Populations of organisms are continually evolving. So all that means is that things are going to change, okay? Things are changing over time. Now, if by any chance you cannot keep up with the notes as I'm moving through the slides, you're more than welcome to pause so that you can write down the notes and then continue to play it once you're ready. All right, so one thing that is not considered to be living is a virus. They're not considered living because they don't share those 5 fundamental criteria. For the most part, they're only going to process information, and they are capable of reproducing, but only once they're inside a host. They can change over time as well, but they do not have any cells, and they are not going to acquire and use energy. A theory is an explanation for a general class of phenomena or observations that's supported by a wide body of evidence. And there's 3 theories that form the framework for modern biological science. First being the cell theory. So what are organisms made of? Number 2 is a theory of evolution by natural selection. So where do they come from? And the chromosome theory of inheritance is how is hereditary information transmitted from one generation to the next. Moving on to part 2. So life is cellular and replicates through cell division. So in 1665, we had a person by the name of Robert Hooke, and he ended up developing a microscope, a very primitive microscope. It only magnified things about 30 times. But what he did is he ended up cutting a small piece of cork. And for those of you that do not know what cork is, it's that little piece of wood that's found inside of a wine bottle. So he took a very, very thin slice of that and he viewed it underneath the microscope. And he saw what this very first image is showing you right there. So he thought that these little compartments kind of reminded him of something. They reminded him of a monastery where monks actually live. Monks have very small rooms and they are called cells. So that he thought that these ended up looking like those small rooms as well. So he ended up terming these little compartments as cells. Then we have a person by the name of Anton von Leeuwenhoek. He devised a microscope that was a little bit better because it magnified things about 300 times. And he ended up getting a small sample of pond water and he viewed it underneath this microscope. And he saw these things right here, okay? They were very small organisms and they appeared to be kind of like animals because they were able to move around. So he ended up calling these animalcules. And then in the 1800s, we have German biologists that claimed that all organisms were composed of cells. So all organisms are made of cells. So in 1660s, Robert Hooke and Anton von Leeuwenhoek were the first to observe cells. Cells were highly organized compartments. They separated their environment they were separated by that membrane barrier. And it formed the first part of the cell theory, where all organisms are made of cells and all cells come from pre-existing cells. So some terminology is that a theory refers to explanations for broad patterns. A hypothesis is a testable statement that explains something observed. An experiment allows researchers to test the effect of factor on particular phenomena. And a prediction is a measurable or observable result, and it must be correct if a hypothesis is valid. Now, one thing to know, guys, is if a hypothesis is stated and tested and it ends up giving or yielding the same results over and over, it can potentially become a theory in time. So there was a hypothesis that existed back in ancient times known as spontaneous generation. And what that is the belief that organisms could arise spontaneously under certain conditions. Like life could literally come from like dirt, for instance. So when the cell theory started to come around, and remember the cell theory is that cells have to come from pre-existing cells. Well, that became a hypothesis. And the hypothesis was that cells are produced when pre-existing cells grow and divide. Now, Louis Pasteur, he ended up conducting an experiment on his own because he didn't believe in the notion of spontaneous generation. He thought that cells did come from pre-existing cells. So we'll go ahead and jot that down. So he ran an experiment where he had 2 glass flasks, and both of them had nutrient broth in them. So basically nutrients that cells could, feed on in order to be able to reproduce. Now, one flask was open to the air, whereas the other one ended up having a swan neck. And pre-existing cells in the air could eventually move into the nutrient broth and grow in the one that was exposed to the air. And it concluded that all cells from Cell's hypothesis was correct. So let's take a little bit of a closer look into the experiment. So here he had 2 flasks, one with a very short neck and one had that swan neck. He had broth in both of them and cells in both of them. And then on step 2, he ended up boiling to sterilize the flask, and it killed all of the living cells that were in the broth. Now on number 3 over here, we see that the pre-existing cells were able to enter the flask from the air, and obviously they're going to end up reproducing, and we see them, in the very last flask. Now over on the other side, the cells from the air were only able to travel so far into that swan neck. They weren't able to migrate all the way into the broth. So obviously at the end of it, we don't see any cells that exist here. So this just goes to prove that, cells have to come from pre-existing cells. So like the cells came from, they were floating around in the environment, they were able to get in and feed on the nutrient broth and reproduce. But here the cells weren't able to get in. So life can't come from non-living things. So that went ahead and disproved spontaneous generation. Life has to come from life. So cells must replicate for life to exist. As predicted by the cell theory, all cells have descended from pre-existing cells. They are connected by a common lineage. So evidence now shows that life arose from non-life early in Earth's history by the process of chemical evolution. Moving on to part 3, life requires information and required energy. We'll just write down life process info. So the chromosomal theory of inheritance was proposed by Sutton and Boveri, and they stated that hereditary or genetic information is encoded in genes. Genes are units located on chromosomes. In the 1950s, chromosomes are molecules of DNA. And DNA is going to be considered the hereditary material, and genes are segments of DNA that code for cell products. Double helix. So each strand of the DNA is made up of 4 building blocks. We have adenine, thymine, cytosine, and guanine. Adenine always pairs with thymine, whereas cytosine always pairs with guanine. The sequence of this code is like letters in a word. DNA is going to carry or encode information needed for an organism to be able to grow and reproduce. James Watson and Francis Crick proposed that DNA is a double-stranded helix. The 2 strands of the double helix are going to be joined by interactions between pairs of bases. So as stated before, A always pairs with T and C always pairs with G. And they're going to be held together through hydrogen bonding. And this pairing allows DNA to be copied, and it preserves information encoded in DNA. The central dogma is going to be very important, so we do need to know that. It describes the flow of information in cells. So what that necessarily is going to be meaning is that DNA codes for RNA, and that is going to code for proteins. So all we're doing for the central dogma is we're taking DNA and we're switching it to an RNA language, and then that RNA is going to be switched into proteins that are going to be vital for us. Proteins are going to be what's responsible for giving us our physical traits. RNA and proteins. RNA are going to be molecules that carry out specialized functions in cells. Messenger RNA is read to make proteins. Proteins are crucial to tasks required by cells, and they form structural components to promote chemical reactions. DNA is going to be copied to pass genetic information from cell to cell or from one organism to its offspring. And copying DNA is highly accurate. So what happens when there is a mistake? DNA sequence changes may lead to changes in proteins. The outward appearance is the product of proteins produced. So DNA sequence changes lead to heritable variations that underlie the diversity of life. Chemical reactions that take place inside cells do require energy, and organisms have 2 fundamental nutritional needs: acquiring chemical energy in the form of ATP and molecules that can be used as building blocks to make DNA, RNA, and proteins. So how organisms acquire energy is central to the diversification of life. And this image right here is showing you what ATP looks like, and that's essentially what our body is going to be fueled by. Plants and bacteria, they are considered to be autotrophic. So they can produce sugar using energy from the sunlight. And they use sugar to make ATP or store it in energy-rich molecules. They can use the molecules absorbed by the environment as food. And pretty much anything else is going to be considered a heterotroph. Just write heterotroph, don't worry about the ick. And a heterotroph is going to be an organism that needs to eat to gain their nutrition, such as this butterfly. Part 4, life evolves. So what is evolution? Darwin and Wallace made 2 claims regarding the natural world. They stated that species are related by common ancestry. Characteristics of species can be modified from generation to generation, so Darwin called this descent with modification. Natural selection is going to explain how evolution occurs. So there are 2 conditions that must be met for natural selection to occur in a population. Number 1 is that individuals must vary in characteristics that are heritable, which means that they can actually be passed on to their offspring. And number 2, in a particular environment, those certain versions of these heritable traits help individuals reproduce more than other versions. Certain heritable traits change over time and lead to an increased success in producing offspring. Traits become more common in a population over time. Now, the key thing here, guys, to know is that natural selection acts on individuals. Evolutionary change occurs in populations, so it's a group of the same type of organism living in the same place. Now, these are some definitions that we do need to know: fitness and adaptation. So the fitness is going to be the ability of an individual to produce offspring. Individuals with high fitness produce many more surviving offspring than do others in the population. So an example of fitness, for instance, would be like that polar bear right there. That polar bear is fit to live in its environment because it is camouflaged. So, predators can't really see it in its surroundings, and also by the hair. They have very dense and thick fur that help to keep it warm. If we were to, say for instance, stick a grizzly bear into that environment, then that grizzly bear would not have high fitness because it's not adapted to its environment. And also because it doesn't have high fitness, it wouldn't be able to produce many living offspring. Now, an adaptation is a trait that increases the fitness of an individual in that particular environment, such as with the polar bear. The polar bear being camouflaged and also having very thick and dense fur to keep it warm in its environment. Those are great adaptations for that polar bear. Okay, moving into part 5, the tree of life. So what is the tree of life? It's going to be a family tree of organisms, and it's going to describe relationships among species with a single ancestral species at its base. So essentially what that means, guys, is that life had to come from one thing, and then eventually life ended up branching off and forming different types of organisms. Phylogeny is the actual genealogical relationship among all organisms. All right, analyzing genetic variation. How is genetic variation analyzed? So biologists are going to study RNA and DNA from different organisms. And what they're going to do is they're going to look at their sequences of adenine, thymine, cytosine, and guanine. So the fewer sequence variations or the fewer differences between two species, that's going to indicate that they're very closely related to one another. Let's take a look at this image down here. So we are comparing the human, chimp, gorilla, and orangutan. And all we're doing is we're looking to see if there's matches here on those sequences, okay? So if they have more sequences that match up, that does show a closer relationship between the two organisms. And if we look really closely, the human and the chimp only have that slight difference right there, and everything else is exactly the same. So that just goes to show that the human and chimp are more closely related than like the human and the gorilla, because the gorilla has more variation. There's differences right here in the gorilla versus the human, and also right within this region and the human. And the orangutan is just completely different from all the rest of the 3 organisms that are mentioned here. Let's take a look at this example. Okay, so all we're doing is we're looking at the nitrogen bases and seeing which ones have fewer differences between the two. So let's compare, first of all, the land plant and the green algae. So we have the A here that matches up, the T, the A, the T. Now the only difference is going to be right here. Okay, the C and the G are different, but here we have similarities, similarities again, and similarities again. Now, green algae and brown algae, let's take a look at them. So right here we're the same. Here's our first difference, so that's number 1. Same, 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 and now they have 2 differences. This is number 2. So in comparing, we know that the green algae is going to be more closely related to the land plant than to the brown algae, because with the land plant, they only had 1 difference, or 1 sequence that's different, and with the brown algae, they had 2 sequences that were different. All right, in this slide, it's going to talk about the phylogenetic tree of life. So go ahead and start with phylogenetic tree, and it's used to show the relationship between species. So branches that share a recent common ancestor represent species that are closely related, such as if we look over here on domain Eukarya, we have slime molds, fungi. They're very closely related to one another, and that's why they're very close to one another on that phylogenetic tree. Branches that do not share recent common ancestors represent species that are more distantly related. So say for instance here, when we look at animals, animals are going to be very distantly related to, say for instance, um, different types of bacteria, and that's why they're further away on that phylogenetic tree of life. Interpreting the tree of life. So the tree of life does include three major groups of organisms. The very first one are the eukaryotes, and we want to think of it as eu, true, pro, no. True eukaryotes do have a true nucleus. Pro, no. Prokaryotes do not have a nucleus. Okay, so eukaryotes have a nucleus, and then we have domain eukarya, and then we have two groups of prokaryotes. So once again, pro, no, they lack a nucleus, and they are going to be bacteria and archaea. Now, bacteria are pretty much going to be found absolutely everywhere. They're common, whereas the archaea are going to be found in areas where there's like very extreme environments. So if you want to think of like hot springs are extremely hot and not many organisms can live there, but we can find certain archaea, which are a type of bacteria that live there. We can also find archaea living, near volcanoes. So these organisms are found in those like very extreme environments only. Taxonomy, that is going to be an effort to name and classify all organisms. So that's just a branch of science that deals with that. Taxon is a named group. The domains we just talked about right now, which are the three taxa, are bacteria, archaea, and eukarya. And phylum is a major lineage within a domain. Now, this image is showing you an example, okay? So in taxonomy, we want to make sure that all organisms do have a name. But unfortunately, organisms do have many different common names, and it all just depends on the area where people live. So like, say for instance, if we were to see this organism in El Paso, we would know that as a mountain lion. But if you go to California, for instance, and you show them a picture of it, they might call it a puma, they might call it a panther. So it gets very confusing when using common names. But everything does have a scientific name, and that's known worldwide. So this organism's scientific name is called Felis con color. In the 1700s, we have a person by the name of Carolus Linnaeus. We'll just write down his last name. And he established a naming system that we actually still use today. And each organism is given a two-part scientific name known as the genus and the species. The genus, actually many organisms can share that same genus name, so it's just a group of closely related groups of species. Whereas a species name, only that one organism can have that species name. So say for instance, Homo sapiens. Homo sapien is a scientific name for humans. Now Homo, there could be other organisms that have that same first name, but sapien, no other organism in this world will have that name because that's specific for humans only. So species is just going to be individuals that regularly breed together, or individuals whose characteristics are distinct from those of other species. Now, rules, an organism's genus and species designation is called its scientific name or Latin name. Scientific names are always going to be italicized. The genus is always going to be capitalized, and then the species name is not going to be capitalized. And here is your example of the human, the Homo sapien. So we notice that both of the words are italicized, which means it's slanted. The Homo, the genus, is going to be capitalized, whereas sapien, the species name, is going to be lowercase. And part 6, finally, doing biology. Just put doing bio. All scientists are going to ask questions that can be answered by measuring things. They are going to collect data. And science involves formulating hypotheses, so it's a possible solution to a problem. And then we're going to find evidence that supports or conflicts with those hypotheses. Testing is a two-step process. You need to state the hypothesis as precisely as possible and list predictions it makes. And two, you're going to design observational or experimental study that's capable of testing those predictions. So people wondered why giraffes end up having very long necks, and there were two hypotheses that came about because of this. So the very first hypothesis was the food competition hypothesis. It argues that long necks evolved because those with long necks could reach food that was unavailable for other animals. They could reach food that was really, really high, whereas other mammals couldn't. So the predictions were that the neck length was variable among giraffes. The neck length in giraffes is heritable, so it's passed on by their parents, and giraffes end up feeding high in trees. So we have two people that ended up testing this hypothesis, Simmons and Scheepers, and their third prediction of feeding high is actually not true. Most people think that giraffes will end up feeding very high into tall trees, for instance, but this is the actual position of how they feed. So they don't extend high in order to feed, they actually extend out in order to feed. Now, the other hypothesis with giraffes having long necks, or the reason behind it, is because of the sexual competition hypothesis. So they are stating that they evolved long necks because those long necked males are able to win more fights than males with shorter necks. And if they're able to win more fights, then they're able to end up fathering more offspring. So upon observation, they actually were able to support this hypothesis. And they were also able to say that the food competition hypothesis was not valid. So all hypotheses have to be tested rigorously before, they're actually accepted in the science realm. There was another experiment that was conducted, and this was now going to be on ants, and they were wondering how ants are always able to navigate back to their nest. Now, experiments are a very powerful tool because they allow researchers to test the effect of a single factor on a particular phenomenon. And that's very important, guys, because you can't test multiple things at the same time because you'll never know which one actually produced the outcome that you were looking for. So Whitlinger and colleagues questioned how ants find their way back to their nest after foraging for food, and they came up with what's called the pedometer hypothesis. So ants always know how far they are from their nest, they track the number of steps that they take, and they know the length of their stride. Now, the null hypothesis is just saying the opposite of it. So if the pedometer hypothesis is wrong, stride length and step number should have no effect on the way back to the nest. This image is just showing you that basically, they're going to go out and forage for food, and then eventually they make their way back to the nest. So experimental setup. Whitlinger's group, they manipulated ants into 3 groups after walking from the nest to the feeder. So manipulated ants. Number 1, they gave ants stumps, so legs were cut to form shorter than normal legs. And then they had the normal group, which nothing was done to them. And then they had stilts. So they glued bristles onto the legs to form longer than normal legs on the ants. And then the team then measured the distance that the ants traveled back to the nest via a different route. In this image, you can see we have the stumps, the short legs, the ones on stilts, and then the normal ants. Let's go ahead and take a closer look at their experiment. So what they did is they collected 75 ants, and the ants were supposed to walk a 10-meter distance from the nest to their feeder. But they manipulated 50 of those ants. 25 ended up having the stumps, their legs were cut, 25 were normal, and then 25 had those bristles attached. And then they set them on the path from the feeder and to search for their nest hole. Now, upon return, they noticed that the ones with stilts ended up traveling too far of a distance. The ones that had normal legs, they ended up hitting it right at the 10-meter mark. And then the one with stumps, they ended up traveling too short of a distance. So in result, they ended up finding out that the stumps stopped short of the nest, the normal ants returned to the nest, and the stilts, they ended up walking beyond the nest. So in conclusion, they were able to determine that desert ants use information on stride length and number to calculate how far they are from their nest. And that went ahead and proved that the pedometer hypothesis was actually correct. There are important characteristics of a good experimental design. One of the first things that we have to note is there always has to be a control group. Now, the control group is a standard baseline in an experiment. It stays the same. It doesn't receive the test treatment. So scientists are going to compare that test group to your control group to see if the treatment actually causes a real change. So in short, what you can write down for the control group is a comparison. It's going to take me forever to write it, but it's a comparison. It's not the one that's actually being tested. It's just going to be there to compare to see if the one being tested actually does work or not. So the control group is just going to answer like what would happen if we didn't apply the treatment. So here in our ant experiment, we have obviously different groups that were being tested. We're overall just looking at leg length. So we had stumps, we had stilts, and then we had just normal ants. So in that experiment, the control group would actually be the normal ants. Other things that we need to know is that experimental conditions have to be constant. So if this test was repeated over and over again, nothing else should be changing. It should be run exactly the same. Repeating the test is essential to ensure that you're getting enough data, and you need to use a large sample size once again to get more data. The more data you have, the more you're able to prove that your experiment actually worked and that your hypothesis can be supported. With that, guys, we are done with chapter 1. Until next time.